Welcome to science class. Today we are going to zoom in and study evolution on the smallest scale yet, that of the DNA molecule itself. Humans are around 98% genetically similar to chimps. What does it mean to be 2% difference? And what does it mean to be 98% similar? Does the 2% difference come from us acquiring additional genes that chimpanzees don't have? Does it come from losing genes we used to have? Does it come from pre-existing genes mutating into new forms? The answer is yes to all three. Differences in phenotype arise from changes in DNA, and those changes can occur in many different ways. As we have previously learned, we also have to contend with minor genetic changes having magnified effects as those genes interact together in unexpected new ways. The other thing we know about molecular evolution is that it is both random and predictable. Here, I'll use the Galton board again. I have no idea where each individual ball will end up, but I do know approximately how they will arrange themselves in the end. Similarly, we can't know which genes will mutate or exactly when they will mutate, but over long periods of time, we can calculate how many unique mutations have accumulated. This means we can look at the genomes of two species, see how much they have diverged from each other, and do the arithmetic backwards to see when they shared a common ancestor. Let's get started. The earliest form of life on Earth would certainly have been unicellular and prokaryotic. What we know about all prokaryotes is they have a single chromosome, plus some DNA fragments called plasmids, and relatively few genes. The longest prokaryote genome that I could find is a bit less than 15 million base pairs in length, while the E. coli genome is only around 5.5 million base pairs. That is between 0.14 and 0.43% of the human genome. So never mind the DNA differences we have between chimps, how have us, other animals, and eukaryotes in general accumulated so much more DNA? First thing, life has been around for more than 3.5 billion years. It is simply impossible to understand how much time that is. But it's not impossible to understand that it is enough time for dramatic changes to occur. Second thing, we actually already know where extra DNA and extra genes come from. Prokaryotes exchange plasmids, which increase the genome of the host cell. This is called horizontal gene transfer, which happens in several ways. When a bacterium receives a plasmid, that is a transformation. When a virus introduces the new DNA, that is a transduction. And there are conjugations, where two bacteria physically attach to each other and exchange DNA before releasing. This gives us a source for additional DNA in prokaryotes. But we also know that DNA itself changes over time. There are errors in the DNA copying process. UV radiation can strike a nucleotide and remove it. These mechanisms and others allow duplicated genes to evolve and serve brand new purposes. Make no mistake, the chances are slim and it happens very gradually. Life on Earth was prokaryotic and/or unicellular for 3 billion of the 3.7 billion years it has existed. That's indicative of slow change. But in eukaryotes, molecular evolution can occur much more quickly. Sexual reproduction alone provides a mechanism for introducing new genes in eukaryotes. Cross-species hybrids or breeding among genetically compatible subspecies can combine genes that have never before existed but now do exist in individuals in a population, leading to new characteristics. Chromosomal mutations occur. Genes can be deleted, duplicated, inverted, or translocated. For example, humans have 46 chromosomes, while chimps, gorillas, and orangutans have 48. 2 chromosomes from our common ancestor fused, producing the second chromosome that all species of humans had, and we, as the only remaining members of our species, still have. Sometimes genes mutate in very unhelpful ways. Almost all primates, plus some species of bats and guinea pigs, funny enough, cannot synthesize vitamin C. We have to get it from our diet. In the case of primates, we have the gene that produces the enzyme that helps synthesize vitamin C, but it mutated millions of years ago and no longer makes a functional protein. So this is what we call a pseudogene. In the 20th century, we learned that regulatory genes, especially Hox genes, are shared across all animal species. What these regulatory genes do is they turn on and off at specific times during embryological development to guide the development process. Specific genes turn on to form limbs, specific genes turn on to form our midsection, certain genes turn on to form our nervous system. You get the idea. The assumption had been that whatever genes control for the development of insect legs had to be unique to insects. Later, we learned that this isn't the case at all. The genes that guide the development of legs, or any other body part, in insects are virtually identical to the genes that guide the development of those parts in mammals. Mammals are more complex, and we will have more of those genes, but we share the ones that even fruit flies have. These genes are so similar across vertebrates that if you cut a piece of a mouse embryo where a leg will eventually grow and graft it to a frog embryo, say on its back, the frog will grow a frog leg on its back. Let's compare arthropods. Insects are arthropods who, among other things, have 3 pairs of legs. Crustaceans are arthropods who, among other things, have 5 pairs of legs. Research now shows us that these two animal groups have the exact same gene, called UBX, which is deactivated earlier in the development of insects, thus causing them to develop only 3 pairs of legs instead of 5. Although we can point to nice discrete examples like that one, in reality this is maddeningly complicated. While the difference in one trait between two groups may be how long a gene is active for, in 2 other groups it will be a matter of how many copies of that gene there are. And in 2 other groups it will be the case that one group has the gene and the other just completely lacks it. And again, the majority of traits that organisms possess are shaped by the interactions of several genes. So while we are 98% similar genetically to chimpanzees, we are not 98% similar in countless other ways, because those small differences magnify. For example, while chimpanzee brains are almost structurally identical to ours, just much smaller, chimps cannot learn to speak, and people have tried. Chimps have been raised and vigorously taught to try and speak their whole lives with no success. It appears that there is something about our brain that is wired subtly but crucially different. For the 1,000,000th time, I would like to add that evolution is gradual. It is not typical to see a new species evolve over a conventionally observable period of time. But that doesn't mean that we can't understand how evolution has occurred in the past. Nobody was alive when South America and Africa were connected, but we can clearly see that they fit perfectly, and we know that they are inching apart from each other every year. So what we can do is extrapolate from the evidence we have and estimate when they were connected. Then we correlate this to other known points of data, such as the ages of the rocks in those areas. We can do something similar with DNA. We know that mutations can be silent. Because an amino acid can have multiple codons, a change in a single nucleotide can still make the correct protein. Or a mutation can occur in a non-coding part of our DNA, which makes up the majority of our and other animals' genomes. These so-called silent mutations accumulate between 2 different species at a slow and steady rate. If we analyze 2 species' genomes and see how many silent mutations they have independently accumulated, we can work out how long it took to accumulate those mutations, which tells us when these 2 species last diverged from a common ancestor. This is called the molecular clock. Molecular clock analysis is not simple. Different regions of DNA accumulate silent mutations at different rates, and the time it takes to accumulate, let's just say 100 silent mutations between these 2 species is not the same amount of time as it takes to accumulate 100 silent mutations between 2 other species. But scientists know how to account for these factors and calibrate the molecular clock. And that is where we will end with evolution. We covered a huge range of topics and ideas. But evolution is the cornerstone of biology, so it was kind of necessary. I hope you were able to learn a lot and enjoy learning. Thank you so much for watching. I'll see you next time.